So in this video, I'm going to talk about multicellularity in plants as it relates to evolution and development in plants. As with animals, most plant species are multicellular. And so they have a number of control mechanisms such as homeodomain genes and genes similar to that control this crosstalk between cells and allows them to develop into organized multicellular complex organisms. Now I want to switch over to plants, the Archaeplastida, which also have homeobox genes and have differential ways of expressing genes to form organs. And in a general sense, they're analogous, but then specifically, the mechanisms are somewhat different. This is the plant clade branching off, and the white sections of this down here, these are primarily unicellular organisms, and then all the dark areas are the multicellular organisms. So at the bottom you have the glaucophytes, and then the next two groups up are the red algae and green algae. Which have both unicellular and multicellular members. Going up further, the bryophytes or mosses, and then up further are lycopsids, ferns, horsetails, and then the spermophytes, which are angiosperms and gymnosperms. Among the multicellular organisms, on the lower end, the primary or larger part of the life cycle, these are gametophytes, or the haploid generations, and they have only a small diploid version of the organism of the life cycle. Whereas if you work your way out on this phylogram, then the sporophytes are the major part of the life cycle. So in plants, the trees and the floral, the flowers and larger organisms are the sporophytes, which lead to the gametophyte, which is a haploid tissue that leads to formation of gametes, and ultimately you reform the sporophyte after fertilization. Also in plants, there are two things called isogamy and anisogamy. So with the very simple plants, even the multicellular plants, then all of the cells look pretty much the same. That's isogamy. So even the gametes, there's no differentiation between the essentially male and female, but they're just called plus and minus sexes. Whereas when you get to anisogamy with the larger organisms, then there begins to be some differentiation between the gametes, so you can tell the difference between a male and a female cell. By the time you work your way up to trees and higher plants, then there's a real large difference between the male and female organs, as well as the male and female gametophytes and gametes. Here's a typical plant cell. The nucleus in the middle, the gray areas, is the cytosol, and then there's a vacuole. Because plant cells are restricted by the cell wall, they control some of the pressure of the cell by using the vacuole, although it serves other important purposes as well. Then there's the endoplasmic reticulum, the Golgi, chloroplast, mitochondria, etc. And then there are plasmodesmata that connect the cytosols of the two cells, the adjoining cells. But they are controlled, so there's some movement between there, some communication, which is different than in animal cells that communicate through proteins connecting both cells together, but not the cytosols. So, in some ways, the physiology and interaction of the cells is quite different from plants to animals. Just as with animals, Drosophila is a model organism, in plants, Arabidopsis thaliana is a model organism. So here is the little plant. It's a little crucifer, tiny little plant that grows fast, goes through its life cycle very fast. There's a flower up in the middle, and then some of the mutants of Arabidopsis are shown on the lower right. On the upper right is a diagram of normal development, and then there's the lower one in B is called the raspberry mutant, and just by a change of a single gene, which is like a domain gene, you can change the entire developmental pattern in Arabidopsis. So that's one of the genes that acts very early during development. The typical angiosperm development, that is flowering plant development, is found, this part is found in the female parts are found in the ovary, which is inside of what's called a carpel, and inside of that is an ovule. Inside of that, you will find what's the female gametophyte. So in humans, right, the gametophyte is just simply the sperm or the egg. In the case of plants, the female has a multicellular gametophyte, and there are typically 8 cells: an egg cell with 2 synergids, 2 polar nuclei, and then 3 antipodal cells. Once the sperm comes down the pollen tube, it is actually 2 nuclei by that time. One is a sperm nucleus, which fuses with the egg nucleus to form the zygote, and then the other one fuses with the polar nuclei to initially form triploid tissue. This is called the endosperm, which is initially triploid, as I said, but quickly becomes polyploid and forms a lot of starch in maize or corn. This is most of what you're eating is that starchy endosperm. The initial cell division of that zygote is asymmetric, and the small cell on top eventually becomes the embryo, and the lower portion becomes the suspensor, which attaches this embryo to the ovary of the plant. Right away, there's differentiation of those cells and the tissue that's formed from them, so that the top part becomes eventually the shoot apical meristem. There's a middle portion that becomes the hypocotyl, which is the embryo stem. And then there's the radicle at the bottom, which is the embryo root. And then still there's that section attaching the embryo through the suspensor to the mother plant or the ovary. Just as in animal development, there are specific genes in plants in the embryo that turn on to produce different types of cells and different types of organs. So up at the apical end, you have PIN1, a specific gene that then eventually helps to form the shoot apex, and then there are genes at the bottom of the plant at the root or radical end that will turn on genes to form that tissue as well. Up on top in the shoot meristem or shoot apical meristem, there's zonation, so you have several layers, L1, L2, L3, and then the P's are leaf primordia that are being pushed up to form new leaves. And then you have a root meristem at the bottom of the plant, which forms the root portions, including the root cap, which is the columella cells at the bottom, which are protective of the root meristem cells. These are the things that push through the soil and are really rugged and tough cells. Here are 2 micrographs just showing the shoot apex at the top of a typical plant, and you can see the shoot apical meristem at the top with the leaf primordia surrounding it. And then at the bottom, you can see the root apex down here. Note that in the root apex, you really have a lot of distinct rows of cells going straight down, whereas in the shoot apex, you do have some rows of cells, but there's also some more bulky cells in the center to kind of broaden out the stem portions of the plant. This is a close-up of a shoot apical meristem, and you can see the leaf primordia on the upper left and upper right, the formation of new leaves. This is actually from my master's thesis on heterohelix, which is English ivy. And you can see in the center here these cells with very large nuclei, as well as smaller cells around the periphery, and more dark staining cells in the middle. And this central part here is called the apical initial zone. And those cells right there actually lead to all the genetic material that forms all parts of the shoot of the plant. So they're really sort of like stem cells of a plant. If you broaden out just a little bit, here's the shoot apical meristem again. You can kind of see darker staining cells in here. You can look at this in several different ways. The first layer, L1, is always one cell layer thick, and that's the epidermal layer, and it's always just one cell layer thick. L2 consists of at least 2 layers, and in the leaf primordia, then those actually broaden out into more layers as you get more and more cell divisions in a different direction. But genetically, those remain distinct, and I'll show you why that is in a few minutes. Another way that people have looked at this is to look at what are called tunica layers and corpus layers. So the tunica are specific layers. There's T1. T2 starts out as one layer, and then you get cell divisions to form more than one layer. T3 also starts out as a single layer, and then you get more cell divisions. But the corpus, which is the central body of the plant, tends to have cell divisions in all directions. Now, if you look at the staining patterns and the sizes of the cells, then you can break it down into zones also. So there's an apical initial zone characterized by darker staining cells. There's a central zone where the cells are more lightly staining. And then there's a peripheral zone where the cells are smaller. They're dividing much more rapidly, and they're, again, a little more darkly staining. So if you look again in detail at this slide, you can see in here there's a little more staining, a little lighter staining out beyond that, and if we go out a little further, the cells get smaller and again are darkly staining. Just as there were homeobox genes in animals, there are analogous genes in plants. One is called VUS, which is one of the initial genes to turn on to control the formation of the shoot apical meristem. And then VOX down below turns on very early to start out the development of the root apical meristem. And up in the shoot apical meristem, there are also some other genes called clavata. And so clavata inhibits VUS, and VUS actually stimulates clavata. So there's a sort of an equilibrium between the two to make sure that the shoot apex grows little by little, but doesn't grow too fast and outgrow the shoot apex itself. On the other end, VOX is controlled by a set of genes in the root apical meristem feeding back to it. The central gene, CRN, is actually stimulating VOX expression, but then VOX is controlling expression of genes down below that to again control the shape and development of the root apical meristem. So once the plant starts growing and forms a true shoot apical meristem, how does it maintain this pattern, and what actually happens to it? What are the L1, L2, and L3? And these were a couple of things that I studied for my master's. So I investigated this for my master's using heterohelix variety gold heart. Gold heart because it has a variegation pattern like this where the center of the leaf has defective chloroplast, so it doesn't form chlorophyll, whereas the rest of the leaf is green. So here's a wild type, here's the normal gold heart, and then occasionally you get leaves where they're only half white and half green on one side, or half green and half white on the other side of the stem. You can relate this back to the shoot apex in that in half of the apex you have a normal wild type cell in one location, and in the other half of the shoot apex, the other cell on the other side, or other groups of cells, have the mutant condition. So in addition to mapping the apex up at the tip of the shoot in these plants, I mapped the green tissue throughout the plant and found this pattern, so that L1 consisted of the epidermal layer all throughout, and then L2 consisted of 2 layers in the shoot apex, but then those broadened out further out. And the green tissue formed the margins of the leaf, whereas the center part, which was the mutant tissue, the white tissue, filled out the center portion of the leaf. So in a wild type plant, all of this would be green, but in the variegated plant, this central part was white. So in this pattern over here, then L1 was 1 cell layer thick, L2 was between 2 and 3 cell layers thick, and L3 just made up the rest. And I could trace back to the center that this was the apical initial zone, and again, these are the cells that lead to all other cells in the plant. So these are more or less the stem cells of the plant. The central zone, other things are being expressed there, and then in the peripheral zone you're getting a greater proliferation of the cells, eventually pushing up leaf primordia and leaves. So there's a general patterning of cell layers called histogenic layers in plants, which are genetically derived layers. Each layer is a specific genotype. But then the next question is, how are leaves born on a plant? How are they set up? And that's actually done by a different set of genes. In plants, if you look at the shoot apex, there's a leaf primordia pushed up in one position, and then another one is pushed up at a later time in a different position on the plant, and then a third one is pushed up, etc. And so you get this pattern of leaves formed in the shoot area of a plant. So if you look down, if you cut across the shoot apex at the level of some leaf primordia, here's one leaf, there's the next, etc. in a whorl. Here's the shoot apex, and a couple of the main genes and proteins turned on are stemless and knotted. And stemless will inhibit leaf primordia until the cells get far enough away from the shoot apex that they're not inhibited anymore, and then they start pushing up a shoot apex, which are stimulated by AS1 and AS2. There are other genes, YAB and CAN, which will determine which is the adaxial side of the leaf, which once the leaf unfolds, it will face the sun, and the abaxial side of the leaf, which will be the underside of the leaf, which will have the stomata for gas exchange of the leaf. When and how these are turned on varies from one plant species to another. So there are many different types of arrangements of the leaves around the stem on a plant. So there's the ones where you have an opposite arrangement, where leaves are born on either side of the shoot apex, 2 at a time. There's decussate, where 2 are in one direction, the next 2 are in a direction that's 90 degrees to that, etc. There are tricussate whorled, which are 3 leaves around one whorl, and then 3 leaves around the next whorl, etc. alternating leaves, which is what happens in heterohelix. There's one leaf on one side, and then further up on the stem there's a leaf borne on the other side, and another leaf on the other side, etc. And then simply whorled, so you have many leaves around each whorl of the stem. Then there are Fibonacci spirals and Fibonacci alternate patterns. And Fibonacci is just a type of spiral pattern. So in each case, these are determined by some of the same proteins formed by some of the same genes, but it depends on the various concentrations of those proteins around the cells, around the shoot apex, that will allow the next primordium to be initiated in a specific spot on the shoot apex. Here are some examples of each one of those. These ones are opposite, so two leaves are formed in each part of the stem. Decussate are ones where you have two going one direction and then two coming out at a 90 degree from those, and then two further up at a 90 degree angle to those. Alternate, this is a heterohelix again, one leaf on one side, then go up the stem, the next leaf is on the other side of the shoot apex, etc. Here are tricussate whorled, where there are whorls of three leaves in each position on the shoot. Generally whorled on the lower left. In this case, many times you can't tell unless you get way down on the stem to determine where the whorls are. And then the Fibonacci spirals and the Fibonacci alternate patterns are shown there too. When a shoot apex changes from a vegetative apex to a floral apex, several things change, including, in most cases, the floral apex is a determinate organ. That is, it's at the end of that stem, whereas a vegetative shoot apex continues growing and putting out new leaves all the way up. But here, several things can affect a transition to flowering, including the effects of different light and wavelengths of light, certain hormones, especially gibberellic acid, and then temperature effects, both warm and cold depending on which plant it is, and then nutrient effects, including sugars and other compounds that affect a transition to flowering. When there is a transition to flowering, these homeotic type genes or developmental regulation genes are involved. One of the ones in the center of this is leafy, LFY in the center there. So once that is triggered, then there's floral initiation. That can influence the expression of this A group down here, A petala, etc. A group called a B group, and then a group over here called the C group, the D group, and then the E group over there. So on the next set of slides, I'll show what the interactions lead to between the A's, B's, C's, D's, and E groups. Here are a few flower types just to introduce the terms of what a flower is. So up on the upper left, this is just a general diagram of a flower. So down at the base of the flower you have the sepals, which often are green but sometimes are colored, and then a whorl around that of petals, and then a whorl around that of stamens, which consist of an anther and a filament. Those are what the anther forms the pollen. And then you have a carpel, which consists of the ovary and then a style and a stigma. And the stigma is where the pollen eventually lands, and then the pollen tube grows down the style into the ovule to fertilize the egg. Flowers can be very diverse. So in B here, this is a lily. So that the petals, there are only 3 petals there with the initials P. The other pink organs there are sepals, so they look somewhat like the petals. And then there are the anthers, and there's the stigma in the center. If you go to C, this is an orchid flower, and they have not sepals or petals, but they're a more primitive flower, and they have what are called tepals, and so those are a progenitor of what would become sepals and petals, but they're a more primitive form of those organs. Indeed, this is a flower from the Rosaceae. This is, I think, a pear tree. There are 5 petals, and you can't see them, but behind those are 5 sepals, and then there are 5 anthers and a stigma. This is from a rose plant, and you can see the 5 petals. Behind that are the 5 sepals, and then multiple anthers and stigmas. And then this is a hibiscus plant over here with the petals, and again the sepals are behind that, and then you have a fused set of a tube-like set of anthers, and then the stigma coming out the top. So quite a bit of variation and diversity in flowers and floral organs. So if you remember the A, B, C, D, and E gene sets from a previous slide, in a normal flower, if you look at the top of that flower, you have, if only A genes are influencing that area, you get sepals. If A and B proteins are produced, you get petals. If C and B genes are produced, you get stamens. If only C's are produced, you get carpels, and in the center, the D genes form just the ovary. And then on the other side, you get a mirror image of that. So for a flower like over here, you have a whorl of sepals, a whorl of petals, a whorl of stamens, a whorl of a carpel, and then in the center, the ovules. In mutants, however, you get different patterns. So here's the normal pattern here. But if you lose the A set of genes, all you get are rows of carpels, stamens, stamens, carpels, stamens, stamens, and carpels. If you are missing the B genes, you get only sepals and carpels. If you are missing the C genes, then you get only sepals and petals. And if you're missing the E genes and their products, then the only things that form in that area are leaves. And so you get, where a flower should be, you just get a sort of bushy looking set of leaves where the flower should be. If we superimpose these gene patterns onto a phylogram of plants, if you start with, typical plants, the rosids and asterids, then you have these common patterns where you get the A, B, C genes. I've left out the D and E genes because they're constant. Where you get a sepal, a row of sepals, a row of petals, a row of stamens, and then the carpel. As you go back further and go into the monocots, especially the grasses, they have very different types of leaves. So there's lemna and palea and lodicule, and then stamens and carpels. And as you go back further into the monocots in the asparaginales and others, you get tepals and stamens and carpels, just as in the orchids. In the orchid family, that's what you get. As you go back into more primitive plants in the magnoliids, the patterns aren't as distinct. So you get sepal-like organs, you get petal-like organs, and you get stamenids and stamens, things that sort of are similar to petals and sepals and stamens, but they are morphologically distinct. So just to show you some of these different types. So in monocots, in a grass flower, such as maize, rice, sorghum, wheat, if you ever go out and look at those flowers when they're blooming, they're kind of indistinct, but you can see them. So there are stamens with anthers and filaments. Those are clear. There's a carpel with a stigma style and ovary. Those are clear. But then the outside floral organs don't look much like petals and sepals, but they are evolutionarily related. So there are lemna and palea outside of that flower, and those correspond to the petals and sepals of a flower from a rose or aster, although they're morphologically quite distinct and inconspicuous. So just as leaves can be quite variable in where they appear on the stems and different leaf types, flowers can be born on the stems in quite a number of different ways, including individual flowers in spike, spikelet, raceme, etc., or in alternating patterns such as the cymes and panicles. Then there are compound flowers. One is called an umbel, and actually you can see those around here quite a bit, and I'll show you one in a few minutes. Corms, there are heads, such as in a sunflower, and I'll show that in more detail in a few minutes. Then there are a few other types shown on the bottom, including the seconium, which is characteristic of figs, where the flowers are in the inside of a little ball with a hole in one end, and in one end the wasp actually crawls into there to gather pollen and lay its eggs, and in the process fertilizes the internal flowers. Here are a few examples. Here's Allium cepa, which is onion, which is in the form of a scape. Here's a raceme. This one, which you see around here a lot, is Daucus carata. This is an umbel. It's Queen Anne's lace. If you see all these white flowers out in areas around, well, almost anywhere, it's a weed. You can see these all around. Each one of those, if you look closely, is an individual tiny little flower gathered together in these compounds. This is the next one down. This is euphorbia, called crown of thorns. And the bright red parts there are not parts of the flower. They're actually modified leaves called bracts. And the flowers are the tiny little things inside. And there are sepals, petals, stigmas, and stamens there. Over here is trifolium, which is clover. And each one of those little purple things is a separate flower and fruit. The next one over is echinacea, and then the one down from here is taraxacum, which is dandelion, and each one of those tiny little things with a petal is a separate flower. Then another compound flower here, which is teasel, and then finally down here helianthus, or sunflower. So echinacea and sunflower have a similar structure in that it has ray flowers around the outside and then disc flowers on the inside. The ray flowers contain no stamens, and they have one petal. They have a stigma and an ovary, and then what's called chaff and pappus, and an achene attaching it to the main body of the plant. The disc flowers, on the other hand, on the inside are all fertile, and so they have stigma with stamens inside of that, petals inside of that, and pappus, which is a modified form of a sepal. Now, if you look at the genes that are turned on in those, then there's a slightly different pattern. The disc flowers have something like a normal floral pattern of the A, B, C, D, and E genes. But then the ray flowers have a different patterning in that the B genes and their products have a narrower range of expression, such that only pappus, which is like sepals, and petals are formed. No stamens are formed, and just a carpel is formed in the middle. So you can get seeds formed in that area, but there are no stamens to fertilize it. Those have to be fertilized by the disc flowers. Okay, so that's the end of this section of development in plants and some of the genes that form the different organs, such as leaves and flowers and the different parts of each. Okay, thanks, and I'll talk to you later.